Thank you. Thank you very much for the introduction. I also want to say thank you to the organizing committee, especially Dr. Kitazawa and Dr. Nochi for the invitation, and of course the Tohoku Forum for Creativity for the great support. Today I will talk about a little bit about my research using beneficial microbes to improve respiratory immunity and increase the resistance to respiratory superinfections through the modulation of the trained immunity. Respiratory infections. Respiratory infections are a big problem for the humankind. Maybe the best example is the past pandemic that we all experienced, so I don't need to convince you that we have to do research on respiratory infection, because despite of the advances we had made in the treatment, for example, in the generation of antibiotics or in the prevention through the use of vaccines, we still have a high mortality associated to respiratory infection, especially in very young children and the elderly. And one alternative to help to reduce the prevalence, or at least to reduce the severity of respiratory infection, is the use of beneficial microbes. In the last decades, a lot of research have demonstrated that the intestinal microbiota can actually modulate the local immune system and increase the protection against gastrointestinal pathogens like a rotavirus, for example. But in the past years, it was also demonstrated that the gut microbiota can influence the immunity in the respiratory tract. In clinical trials and also in animal models, it was shown that the alteration of the gut microbiota can decrease some immunological functions in the respiratory tract and improve and increase the severity of respiratory infection. So now we know that the normal microbiota, that is characterized by a high microbial diversity and a high biomass, quantity of bacteria in the gut, are related to the appropriate immune responses in the whole respiratory tract and especially in the lung. We have also learned a lot about the mechanisms through which the gut microbes can modulate respiratory immunity. One is related to the concept of the common mucosal immune system. We know that all the immune cells located in the mucosal tissues can communicate with each other with the interchange of cells, and also with the release of immune factors like cytokines, interferons, and growth factors. And it was demonstrated that one signal given by the intestinal microbiota is the mobilization of cells like T cells and B cells, and the release of interferon that can reach the respiratory tract and then impact in the respiratory immunity. Another mechanism proposed is the release of the microbial associated molecular pattern. Actually, the previous speaker showed one publication. There is evidence that some peptidoglycan fragments can go through the intestine and reach the blood and directly stimulate the respiratory immune system, or indirectly through the stimulation of immune cell progenitors in the bone marrow. They can improve the resistance to bacterial and viral pathogens in the respiratory tract. And more recently, it was demonstrated that some metabolites produced by the microbiota, like the short-chain fatty acid, that can reach the respiratory tract and then induce what we call a metabolic reprogramming of cells, modulating also the response. And more recently, it was also shown that the microvesicles, this is a kind of fashion now, everybody is doing research in the microvesicle, but they also investigated whether they have some influence in the resistance to infection, and it was demonstrated that actually these microvesicles produced by the intestinal microbiota can reach the blood and be located in the respiratory tract and then induce changes in the immune responses. Although several papers have described different mechanisms through which the gut microbiota can send signals to the respiratory tract, all of them agree that the main effect is related to the innate immunity. in fact, most of the studies demonstrated impact in dendritic cells, both in the conducting airways and the lungs, and also in pulmonary macrophages, especially the alveolar macrophages. And there are also some evidence that even respiratory epithelial cells can be modulated by signals coming from the gut. And through the modulation of the innate immunity, they can also have some influence in adaptive immunity, both in cellular and humoral immune response. So, one of the most important cells that are modulated by the signals coming from the gut are macrophages that have a very important function in the generation and coordination of immune responses in the respiratory tract. And several works have demonstrated the important role of macrophages. This is one of the first published papers that described the influence of gut microbiota in pulmonary macrophages. This is a very simple study. They use mice and they treat it with a mixture of antibiotics, and then they challenge treated and non-treated mice with the influenza virus infection, and you can see here very clear how the treatment with antibiotics reduce the capacity of mice to resist the respiratory infection. And when they evaluated the immune response, evaluated the expression of several factors in the whole lung, and particularly in the macrophage population in the respiratory tract, most of the immune factors, including type 1 interferon and antiviral factors, as well as the balance between pro-inflammatory and anti-inflammatory cytokines, were altered in the mice that received the antibiotic treatment. And there is another study, very interesting study. In this study, they choose to work with, this is laboratory mice, coming from the very famous animal suppliers, and they also work with wild mice. I think they request the PhD student to go and hunt mice surrounding the institute in different locations, and then they compare the microbiota of the mice, and you can see here the very clear differences of the intestinal microbiota between the wild mice and the laboratory mice, and how big is the variation in the wild mice. And when they challenged the mice with influenza virus, they saw that the wild mice were more resistant to the infection than the laboratory mice. And then they designed this very interesting experiment. They used laboratory mice, and then laboratory mice with a depleted microbiota that receive a fecal microbiota transplant from laboratory mice and for wild mice in the green one. And you can see here how the wild microbiota can improve the resistance to the infection, and through a different modulation of antiviral factors and the reduction of pro-inflammatory cytokines and the improvement of anti-inflammatory cytokines. There are several others works that demonstrated the influence of the microbiota in macrophage function. So we know, we have learned a lot about the signals and how the immune response is modulated, but we still don't know which are the members of the intestinal microbiota that are important for the induction of this beneficial effect. We still don't know if it is related to the specific group of bacteria in the gut, like for example, a family, a genus, a species, or even a strain. So there is still research ongoing to find out which are the important bacterial population related to this effect. But I have to say that the researchers working in probiotic, we have done a step forward in this direction, because we already demonstrated that this is a strain-specific characteristic. Probiotics able to modulate the immune system that we call immunobiotics have been shown not only to stimulate the local immune system when they are orally administered, but also they can influence the immunity in distant mucosal sites like the respiratory tract. And in fact, you can find in the literature several strains belonging mainly to the group of lactobacilli or bifidobacteria that can improve respiratory immunity and enhance the resistance to bacterial and viral pathogens when they are orally administered. One of these strains is one lactic acid Bacillus rhamnosus, CRL 1505, that is, was characterized in my laboratory. This strain was isolated from goat milk and was selected in animal models by using adult immunocompetent mice and immunocompromised mice because of its capacity to improve the resistance to pneumococcal infection. And then we move to research if this strain can also modulate antiviral immunity in the respiratory tract. And first, we use a model to imitate the inflammatory response in the respiratory tract. In these experiments, mice were fed this bacteria and then challenged by the nasal route with the toll-like receptor 3 agonist, the poly-IC. And you can see here that the lung damage was significantly reduced, meaning that the strain was able to somehow modulate the toll-like receptor 3 mediated immunity. And when we tried real challenge, actually we saw that mice were more resistant to respiratory syncytial viral infection and also to influenza virus infection. And in this graph, you can see that there is another strain. This is the 1506, this one, 1506 strain that is actually very good immunomodulatory strain. This is, both strains are similar in improving protection in the gastrointestinal tract. Both of them improve the resistance to rotavirus infection, but only the 1505 can induce a stimulatory effect in the respiratory tract. That is why we use always the two strains, because we are trying to understand why one immunomodulatory strain has this characteristic and the other not. We have done a lot of research during the past year trying to find out the mechanism by which this strain modulates the respiratory immunity. I show you these two important pictures. One is related to the study of alveolar macrophages. When alveolar macrophages are depleted by the time of the strain administration, the ability to protect against the respiratory infection is lost. And by using blocking experiment against several factors like interferon gamma, interferon beta, interleukin 10 receptor, and also several type of cell like CD8, CD4, etc., we found that this strain is able to stimulate a Th1 response in the gut and then induce the mobilization of CD4 positive cells able to produce interferon gamma. And once they reach the respiratory tract, they can stimulate antigen-presenting cells like dendritic cells in the conducting airways and also alveolar macrophages. And when these cells face a challenge, like a bacterial challenge, like a Streptococcus pneumoniae, or a viral challenge, they produce higher amounts of interferon gamma and interferon beta, inducing a reduction of the replication of the virus. And we also show that the macrophages can collaborate in the regulation of the inflammatory response through the production of two important cytokines, interleukin 6 and interleukin 27. They can act on regulatory T cells and stimulate the production of interleukin 10, and all these cytokines collaborate to protect against the inflammatory damage during the course of the, for example, respiratory syncytial virus or influenza virus infection. So all these experiments were done in animal models. So we wanted to know if actually this strain worked in human. So we performed a very nice clinical trial by studying the effect of this strain in children from 1 to 5 years. This strain was administered to the children in a yogurt. And of course we included a control group that received only yogurt, a placebo control. And this strain was administered during 6 months, including the winter season, that is because of the high incidence of respiratory infection, and also the summer season because of the high incidence of intestinal infection. And you can see here that the incidence of infection is significantly reduced in the group that received the immunobiotic strain, and this was related to reduction in both intestinal and respiratory infection. And we also detected that in this group of children that received the strain but was infected, the severity of the symptom was also reduced. So We transfer this strain to a very nice company that can distribute the strain all around the world. In Argentina, you can find this strain in this product, and I was happy to hear about Dr. Kitazawa that attended last month a conference in Argentina that you can also find this strain in the product in Japan. And actually, you can find in many other places because we have done maybe 10 years of a study evaluating the antiviral immunity of this strain. Suddenly, when the pandemic came, it became very popular because actually it can help against respiratory viral infections. But we didn't stop our research there. We next moved to investigate whether this strain can do something in the context of the respiratory superinfection. The respiratory superinfection are produced generally by a primary viral infection followed by the secondary bacterial infection. In adults, the most common association is influenza virus, followed by the Streptococcus pneumoniae, but in children, the most common association is respiratory syncytial virus, followed by Streptococcus pneumoniae. This is important because even these superinfections are not high in incidence. They put in serious risk the life of the children. A very high percentage of the children suffering from this superinfection are actually developing very severe respiratory disease. The virus can affect the normal function of the lung. They also produce immunopathology, and it was also demonstrated that they can interact, and then the virus can stimulate a transcriptomic response in the bacteria, increasing the expression of virulent factor. So this bacteria in this context can become very deadly for the infected children. So to evaluate the effect of the strain, we developed this model of respiratory superinfection. We gave the infant mice the strain by the oral route, and then we stimulated by the nasal route with the poly-IC to imitate the toll-like receptor-free inflammation in the lung. And 5 days after the last poly-IC administration, we challenged mice with pneumococci. And as you can see here, we evaluated the resistance to the infection and the damage of the lung, and you can see that there is a reduction of the lung colonization and the parameters that evaluate the lung damage. And most importantly, the strain was able to avoid the dissemination of pneumococci into the blood. That is one of the main characteristics that put in risk the life of infected children. And when we did the same experiment, but not with poly-IC, but using respiratory syncytial virus, we got the same results. We also wanted to know if other strains with immunomodulatory capacity can exert a similar protective effect. And we tried several strains of the same species, and you can see here that only this one, this is selective Lactobacillus plantarum, MPL16, was able to increase the resistance to the infection, reduce the lung damage, and also modulate the immune system in the same way of the 1505. You can see here that we have two different species, and immunomodulatory strain of the same species cannot cannot achieve that effect. So again, we can say that this is a strain-specific characteristic, but it's not related to a specific species. Then we focus on the alveolar macrophages. And we studied after the poly-IC administration, 5 days after, we took the alveolar macrophages and made primary culture, and then we challenged in vitro with the Streptococcus pneumoniae. And in this experiment, we replaced the 1506 by the MPL16 strains for the plantarum group, and you can see that most of the cytokines, the interferon, the inflammatory cytokines, and interleukin-27 are produced in higher concentration in the animals that were treated with the immunobiotics, but not the interleukin-10. and this effect is not achieved by the other strains. We also evaluated the expression of the MHC2. This is an activation marker. There is no great differences in the total number of alveolar macrophages, but the activated macrophages are higher in the groups that receive the immunobiotic strains. These two characteristics, the higher production of inflammatory cytokines after the secondary challenge, and also the high expression of MHC2 in macrophages have been associated to the development of trained immunity. And another important characteristic of the trained immunity is the long-term protection. So we wanted to know the time that mice were protected from the secondary challenge. So we did a similar experiment, but in this time we challenged the mice at day 5, 10, 15, and 20 after the last poly-A administration, and we evaluated the resistance to the infection, the lung damage, and the immune response. As you can see here that up to 20 days we have the same effect. It means that the treatment with both immunobiotics were able to confer a long-term protection against the secondary pneumococcal pneumonia. We also evaluated the kinetics of the immune response. In these experiments, mice were treated with the bacteria, challenged with poly-A, and then 20 days after the last poly-A administration, challenged with the bacterial pathogen. And you can see here that the macrophages and neutrophils, and also these two inflammatory cytokine and chemokine, has different kinetics. They are produced in higher concentration and levels early in the first hours post-infection, and then they tend to reduce in the latest hour. This is very different compared with the control. So the kinetics of the response is also modified by the treatment. That is also a characteristic of the trained immunity. And this effect, again, is not achieved by the other strain. So considering the concept of the trained immunity, that consider that the secondary response of the trained immunity phenotype acquired cell, like the alveolar macrophages, we have demonstrated this one, that there is an increase in effector function, and also there is a long-term protection. But there are two other important characteristics that must be studied to define if a cell actually acquires a trained immunity phenotype, that are the epigenetic modification and the metabolic reprogramming. So we are now studying this one in Argentina. One of my postdoc students is doing this experiment to study if there is some metabolic changes in alveolar macrophages. And here, my assistant researcher came as a GSPS postdoc, and he will do this experiment in the laboratory of Dr. Kitazawa. And then we will know, demonstrate effectively that the immunobiotic strain actually induce trained immunity in alveolar macrophages. So I show you this mechanism at the beginning of my talk. Then I can now complete this mechanism by showing that after this primary viral infection, the benefits induced by this strain in the context of the primary viral infection, they can induce or should induce alveolar macrophages with the trained immunophenotype, and then can confer a long-term protection against secondary pneumococcal pneumonia that is characterized by the improvement of interferon gamma that help to the reduction of the colonization in the lung, interferon beta that is strongly associated to the cytokine avoid the dissemination of the pneumococci into the blood, and also the improvement of this cytokine that may help to protect against the lung damage in the gut, the lung damage against the bacteria. So it is possible to use immunomodulatory lactic acid bacteria by including them in functional food or feed to regulate the respiratory and systemic antiviral immune response, prevent the inflammatory damage, and now we know that it is possible also to reduce the risk to secondary bacterial infection. And this effect is achieved by the induction of trained immunity in alveolar macrophages in the respiratory tract. And this is me thinner. And I want to say thank you to all my students in Argentina. My laboratory right now is full of postdoc. I am searching for PhD students. And also the group of Dr. Kitazawa that have a great hand always with my research. So thank you very much.